All right, this is your introduction to Chapter 7, which as you can see is called Membrane Structure and Function. So in case you are wondering, this is a picture of a one-celled organism, and it's one that we call a paramecium. It is eukaryotic because it has a nucleus. It also has all these little hair-like structures on the outside called the cilia, which from your Chapter 6 reading, you may or may not remember are made of microtubules. All right, and but this whole chapter is devoted to this outer membrane here, the plasma membrane, which you might recall from class is known as the guard gate of the cell. And here we have a picture. This diagram you will see is found on page 29 of your chapter guides, and part of your homework is to label all these different structures. Well, maybe not all of them, but just look for the list on the top of the page. So this will be your homework assignment to label this diagram. All right, I don't want you to write this down. I just want you to listen. The sort of guiding ideas of Chapter 7 are as follows. And then when we're done with the chapter, just go back and all three of these should make sense. So the guiding ideas are as follows. The structure of the plasma membrane determines what substances can or cannot easily cross it. And this should make total sense to you after we've done this whole chapter. All right, small molecules enter and leave the cell by either passive transport or active transport. And finally, larger molecules or bigger items enter or leave by endocytosis or exocytosis. All this should make sense to you at the end of the chapter. Okay, the structure of the plasma membrane. We've alluded to this a little bit before. All right, you might remember that we identified the plasma membrane as a very, 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 very thin layer of lipids, so thin it's only 2 molecules thick. And in fact, to be more specific, it's not just a lipid bilayer, it's a phospholipid bilayer. And the metaphor we use, or the model we use to describe this, is called the fluid mosaic model. So, if you've ever seen a mosaic, it's a picture made of lots of little pieces. So the proteins are the mosaic. And I forgot to mention this, I apologize. You want to be looking at page 33 of your chapter guide. 33, because that's really a lecture guide for this lecture, and we're just going to go over the first part, but you might want to be taking even a few more notes than what I have on here. But on the lecture guide, you'll see we are talking about the fluid mosaic model. So the fluid part are the phospholipids. All right, the fluid is the sea of lipids here, the sea of phospholipids. And then the mosaic part, as I've already mentioned, are the proteins. So these proteins are just literally floating around in the sea of lipids, which is the fluid part. All right, so as I often like to say, you're not allowed to forget anything in biology, and you should know what fats, which are also known as triglycerides, are composed of. So hopefully you remember from Unit 1 or Chapter 5, to make a fat molecule you need 1 molecule of glycerol and 3 molecules of fatty acids. Let's do a little review here. You should recognize this functional group. What is it? Are you thinking hydroxyl? I hope so. And what about the fatty acid tail? Well, anything that's called an acid in biology needs the organic acid group, which is also known as the what? Carboxyl group, yes. So we have the 1 molecule glycerol. Here you see 1 fatty acid tail, and the OH and the H elope. Water leaves. Now we have this bonding together. So again, this whole process is called dehydration synthesis. And not only we do dehydration synthesis to combine 1 fatty acid tail to the glycerol, we have to do it 3 times. So this whole thing, which I see is a little bit cut off, is one great big disgusting fat molecule. All right? So here you have these 3 carbons, which are unnaturally spread far apart in the diagram, and then you have a fatty acid tail, which will be like 16 to 24 carbon atoms long. All right? All right, another little review. You should recognize and see whether this fat molecule is saturated or unsaturated. Want to give a guess? Hopefully you're thinking saturated because there's all single covalent bonds between the carbon atoms. Now some of you, unfortunately, on a test or a quiz incorrectly said a saturated fat has 0 double covalent bonds. That's not entirely true, right? There's a double covalent bond O left over from the carboxyl group. But it is correct to say that there's only single covalent bonds between the carbon atoms, or no double covalent bonds between the carbon atoms. All right, so again, this is a little review. You should be able to identify the differences between a saturated fat, again, we've already said, contains as many hydrogen atoms, not bonds, as many hydrogen atoms as possible, and all single covalent bonds, and you might remember the straight tails, no kinks. Whereas the unsaturated fat, you have at least one double covalent bond between carbon atoms, and wherever you have a double covalent bond, you're going to have a kink in that fatty acid tail. So those unsaturated fats, those oils, are kinky. All right, and since this has more than one double covalent bond, it is polyunsaturated. So that is total review for you, I hope. But now we're going to talk about phospholipids because the cell membranes are the cell membrane is a phospholipid bilayer. So this is sort of funky. Here we see the one molecule of glycerol. We see one fatty acid tail. We see two fatty acid tails. And notice we have a saturated one and an unsaturated one, which is totally legal. But instead of a third fatty acid tail, we have this phosphate group, this PO4 with a highly negative charge. And there's this little moiety up here we're just not even going to worry about. All right, well, the phosphate is attached to this third carbon and it's on the opposite side. Why? Because these fatty acid tails are fats. They're hydrophobic. They're water-hating. But that phosphate is highly negative, loves to dissolve in water. It is hydrophilic. So that is why the phosphate is on the other side of this central glycerol molecule. And every biology in the world does not like to draw phospholipids like this because it's too much work. Instead, you'll see a drawing with, it looks like a balloon with two fat tails. So to review, here's the phospholipid, here's the glycerol, here's the two fatty acid tails, and the phosphate group right here. And again, no biology in the book in the world draws this. Instead, they draw a phospholipid like this. So the balloon, what's represented as the balloon is the hydrophilic head. That has that highly negative phosphate. And then these two strings are the hydrophobic tails. Now again, even though every biology book does this, it's sort of dorky because remember glycerol is only 3 carbon atoms wide. And if that's 3 carbon atoms, well, this whole fatty acid tail is like 16 to 24 carbon atoms. So it's not drawn correctly, it's not drawn to scale, but again, every biology book in the world does it. FYI. So back to our plasma membrane. Here we have all these incorrectly drawn phospholipids, all right, or I'm just telling you. And here you have a whole line of phospholipids in a lion. Do the can-can. All their legs are on the same side. And there's a second layer of phospholipids upside down on the other line, also doing the can-can. And I don't believe this here that the tails just have a divide. These fatty acid tails, they integrate with each other. They play footsies with each other. So this tail might go all the way down here and all the way down here, and this goes all the way up and all the way up because this is all hydrophobic and they all just love playing footsies with each other. So why is this good for a cell? Well, the inside of the cell is cytoplasm, and the cytoplasm is the watery goop or jelly of the cell. Yeah, I do not have my good stylus. I apologize. So the inside of the cell is the watery cytoplasm. The outside of the cell is watery. It could be that one-celled paramecium you saw on the far side. This could be one of your body cells and your blood is watery. But at any rate, with the plasma membrane being a phospholipid bilayer, everybody's happy. All right, the hydrophilic water-loving heads face the outside, which is watery, and the inside, which is watery. Now we just look at the proteins, right? Because, again, this is the phospholipid bilayer. This is a fluid mosaic model. All right, so this is what people tend to forget. What is a protein here? Remember, is really a chain of amino acids, right? All folded up. You're seeing its tertiary structure here. All right, so now all the amino acids in this part of this protein, here, let me draw it over here. They must be water-loving because they are facing the outside water and they are facing the hydrophilic heads here. So this part of the protein is hydrophilic. But this part of the protein, which is surrounded by all these fatty acid tails you do in the can-can, this must be hydrophobic or water-fearing because it's surrounded by fats. So all the amino acids folded up in here must be hydrophobic. Get it? All right. So Again, do not forget, this whole model is called a fluid mosaic model, where the fluid is the phospholipid bilayer and the mosaic are the proteins that are embedded. Okay? And this protein, again, on top, it's popping out, but you're assuming there's a part of it that at least goes part way down. Okay, there's a vocabulary word I need you to teach you, and that is amphipathic. Amphi means both. Think of an amphibian. An amphibian, like a frog, it lives in both the water and the land, right? So amphipathic, and I apologize it's cut off, it's supposed to say both a hydrophobic and a hydrophilic region. So an amphipathic molecule has both a hydrophilic, here we go, a hydrophilic and a hydrophobic region. So every single phospholipid is amphipathic because it has a water-loving part and a water-hating part. But what about the proteins? Are a lot of the proteins amphipathic? Yes, they are. If they go all the way through the plasma membrane, or even part of the way through the plasma membrane, they are amphipathic. But this little guy here, whose name you haven't learned yet, he would not be amphipathic because he's totally in the water. And I apologize for not being able to draw a good arrow. So I'm going to end this little mini presentation with a little metaphor. So again, here we have one layer of phospholipids doing the can-can and another one underneath. So let's do a human metaphor. Here's a whole bunch of people pre-COVID, not socially distanced, side by side, waiting for the bus. And the umbrella would be what part of the phospholipid? Well, that would be the hydrophilic head, because the umbrella loves water. It doesn't matter at all if it gets rained. But the humans are all afraid of water, so their two legs are like the hydrophobic tails. So this is our phospholipids, the hydrophilic head. They love water, the two hydrophobic tails. But if we really wanted to make a plasma membrane out of them, we would need another layer of humans, all right, waiting for the bus underneath. Or maybe it's just the reflection of the water. All right, so I'm going to end here. You're going to do page 30, I believe, for homework, and you get to look at some of these fun facts about how the phospholipids move. And here's a little help for your homework. Do you see this molecule here embedded into the phospholipid bilayer that has 4 rings? That steroid shape? That's cholesterol, FYI. All right, I will see you in the next video.